Hey guys, so today we're picking up where we left off, talking about what happens in mitosis, things that we can expect to happen, and then what happens when mitosis starts to go wrong and cells take that unfortunate step towards becoming malignant or cancer cells. So we're starting off by revisiting the idea that not all cells are destined to divide. Not all cells are consistently entering S or G2 or M. Some cells will stay in G1 or what we call a G naught or a G0 phase so that they're going to repeat this regular living your best life section. Okay, so some cells like this neuron up here, so you can see the axon, which is this bright part coming down here, and then we can see the cell body, and then all of these little green kind of hairs that are coming off are dendrites. And so what's happening is that this neuron grows these dendrites and creates these connections throughout an organism's development. Well, by the time an organism is fully developed into their adulthood, many of their neuron connections look like this. They are very, very complicated. Now, if you imagine this cell needing to recreate that whole series of networks, it would not be possible. The cell would not be able to do it. So cells like neurons don't continue dividing. Once they make it to adulthood, to their fully differentiated adulthood, they are actually going to just stay exactly how they are. Other cells, like skin or blood, are going to be constantly dividing, changing almost every time they make it to the end of G1. They get big enough, they've done their job, they're going to just jump right into the S phase and round again into mitosis. So, what a cell does, it just really depends greatly on what type of cell it is and what kind of connections it has made throughout its lifetime. When we control cell growth, that process is fairly complicated. It's not just a single thing that's going to determine whether or not a cell is going to divide. There are a number of factors that all need to work together in order to start or stop cell division. This kind of overlapping oversight is set forward to try to help the cell make sure that it doesn't divide if it shouldn't divide. Cells dividing at too high a rate is predominantly something that causes cancer or just negative things, other tumors within the organism. So we try to make sure our cells only divide when it's appropriate for them to do so. There are a number of factors that contribute to whether or not a cell is going to divide. One of those is called a growth factor. So a growth factor is a protein. It's a class of proteins. That means there's lots of them that could be growth factors. It's a class of protein that says it's time to divide. So you could have a growth factor as you are growing up. So when you're going from an infant to a toddler, to a child, to a teenager, to an adult, you're going to see growth factor being produced. You can also see growth factor, like we talked about before, if you have a cut. So if you have an abrasion, an opening in your skin, then those cells might be notice the opening, notice the space, the airiness, and they may decide to start secreting growth factor to tell those cells, well, it's time to divide. So growth factor tells a cell that they should divide. Cyclins are also needed in order to push forward cell division. And so cyclins are seen all throughout the cell cycle, and they kind of boost the cell into the next phase. When you're moving from G1 to S, or S to G2, or G2 to mitosis, there are cyclins involved, which is a group of proteins different than growth factor proteins. Cyclins help with the cycle, right? They help push the cell through the cycle. One thing that we tend to see in cancer cells, in cancer tumors, is an overproduction of cyclins. So even though there maybe isn't any growth factor telling the cell to divide, the cyclins are still kind of shoving the cell through the cell cycle, which is creating more and more cells where they aren't necessary or they're possibly not healthy cells. The last part of this is something called apoptosis. Apoptosis is programmed cell death. Cells are supposed to divide for a certain number of divisions. Depending on their lineage, depending on what kind of cell they are, that number could vary. But they're supposed to divide for a certain amount of time and then they're supposed to stop. They're supposed to die. Because if they keep going on, they've probably accumulated too many mutations over each cell division. So every time a cell divides, it gets some mutations. And after a certain number of divisions, that cell conceivably would be damaged. And so we don't want them to keep going. Too much, though, of this apoptosis, programmed cell death, is seen in AIDS, acquired immunodeficiency syndrome, and also in Parkinson's disease. So too much cell death is a problem. Too much cell division is a problem. We really need to maintain this homeostasis to keep the organism healthy. We need just enough division, but not so much. Kind of like the growth rate, if you want to think of the human population, you would need to have the right number of humans being produced and not too many dying in order to keep our growth rate consistent and healthy. The same is true for cells. So this is a look at the cell cycle again with the checkpoints that we know exist. So starting up here, we always start at the beginning of G1 because that's when the cell is born, like that's its origin. So at the beginning of G1, the cell lives its best life, right? It's going through its regular, everyday process. And some cells, like muscle cells and nerve cells, are going to hold in this G1. And we actually say that they enter this G0 or G naught phase, and they kind of keep cycling in this just everyday metabolism. However, some cells are going to enter at this restriction point, the start. It's the start because when you cross this line, you're starting the process to cell division. And so crossing this line is influenced by growth factors. So is it time to divide? Nutrients, are you healthy enough? Cell size, are you big enough? And DNA, are you healthy enough? Back to healthy. Is your DNA correct enough to sustain division? If the answer to all of those is yes, then the cell can enter into the S phase and through the G2 phase. And at the end of the G2 phase, we check again. And we say, okay, so did everything go okay? Did we grow big enough to divide into two? Did we replicate our DNA with enough efficacy, enough correctness that we feel confident in making new cells? So if the DNA was replicated poorly, or the DNA appears damaged, or the cell is too small, then we may turn around and kind of hang out here in this G2 until we make the corrections. Or apoptosis, we may end up killing the cell off. We may say, you know what, there's too much damage here to your DNA. You're really not healthy enough to be reproducing. We're just going to kind of get rid of you to leave room for extra healthy cells. Remember, they're all part of the same organism. So we're not like killing the organism. We're just selecting for healthy cells within the organism. Assuming that everything has happened right. So we've replicated the DNA with efficacy correctly. We have increased our cell size to the appropriate size. Now it's time to enter the mitotic phase. So now we go through cell division. And there's one last checkpoint, and that's at the metaphase anaphase transition. So you should have in your minds this idea: metaphase, we're lined up at the metaphase plate, and then anaphase, we're going to pull apart. At that metaphase anaphase transition, there's one more check to make sure that all of our spindle fibers have attached, that all the chromosomes are there, all the DNA looks healthy, everything looks good to go, and then we go ahead and let the cells divide. So a damaged cell should not be able to make it through all of these checkpoints. P53 is a gene involved in this first restriction checkpoint. And when P53 is damaged, cells will enter into the S and G2 phase even if they're not healthy, even if they don't have growth factor, even if their DNA is damaged, they're going to jump this checkpoint. And So what we see is in almost every cancer cell, P53 is damaged. That doesn't mean that a damaged P53 causes cancer. What it does mean is that in order for cancer to take hold in the body, P53 needs to be damaged. So, something that we notice all cancer cells have in common is this error in the P53 gene, which allows them to jump from G1 into S without meeting these requirements of healthy nutrient, cell size, and appropriate DNA. They can jump across into the S phase even if their cell is damaged. So you have probably heard of cancer before and have an idea of what it means to have a tumor. So when a cell undergoes uncontrolled cell growth, it is skipping those checkpoints which were previously listed. It is not entering G0. It is not checking for health before it divides. It is just constantly dividing at too high of a rate. We are creating a tumor at that point. So that tumor can either be malignant or benign. And so when someone has a tumor, they often go in to get a biopsy and everyone says, oh, we hope that it's not malignant. We hope it's just a benign tumor. What a benign tumor means is that even though the cells are dividing too quickly and they're definitely in the way, they are not actively trying to choke off and kill the good cells. When a cell is malignant, when a tumor is malignant, that means that those cells are termed cancer. And not only are they growing too quickly, but they are also aggressive. They're looking to kill off the good tissue in the area, and they are capable of metastasizing. And so metastasize means that they break off and they land somewhere else in the body and make little satellite tumors. So if you had, let's say, breast cancer, you might see some cancer cells in the lymph. If you also have cancer cells in the liver, then we would say, well, that was a metastasis of the breast cancer. So the breast cancer went to the lymph and then traveled to the liver and is now living in the liver. It's still considered breast cancer, but it's breast cancer in the liver, or breast cancer on your brain, or breast cancer in your bones. And so those are all places where breast cancer can metastasize. And so when someone gets diagnosed with breast cancer, the first thing that they check is it in the lymph? If it is in the lymph, then they go ahead and check, well, where else is it? And so they do a full body scan to look to see if that cancer has metastasized. They then stage the cancer based on the progression of the cancer tumor. If it's localized in one place, that's considered to be stage 1 and small. If it's localized in one place but it's large, that's stage 2. Stage 3 means that it's been crossing over its borders, that it's maybe digging into other tissues. It's going to be harder to remove. Stage 4 means that it's all throughout the body. It's already metastasized. And so the lower the stage, typically, the better your cancer prognosis is. So when you hear people talk about it, that's kind of what they're talking about, is basically what is the prognosis. The lower the stage, the closer to 1, the better the prognosis or the chances of survival are. So since we're speaking about cancer, I thought it would be prudent to cancer. Cancer is one of those amazing things where the kind of treatments that we have for it are always changing. The amount of information, research, and development that's going into how cancer is treated is kind of off the charts. I mean, by the time something is published, like a book, it's outdated. There are so many new treatments that are coming out for this giant disease. A few options for cancer treatment. The first one is a surgery to remove as much of the tumor as possible. Sometimes this is the first thing we do. Sometimes this is the last thing you do. Typically, radiation comes first before surgery, but sometimes it comes after. It depends. The oncologist, which is the doctor that treats cancer, the oncologist is the one who will make all of these decisions. So, for example, currently, if you're diagnosed with breast cancer, your first line of defense is actually going to be chemotherapy. You're going to do a targeted treatment that's going to attack the cancer cells of the tumor. You would follow that up usually with a surgery to remove as much of the tumor as possible so that you can exactly map where the tumor was. And then you would end the whole process by doing radiation, which is going to, again, target the tumor and any of the lymph tissue or other locations where you think that the tumor might be. It's going to target those directly and irradiate them, which is going to cause them to die, those cells to die. For every treatment of cancer, the hardest part about it is that it is your cells. And so it's really difficult for your immune system to fight your own cells or to recognize your own cells as being the problem. And so that's why we need to use these other outside targeted treatments instead of enhancing your own immune system. For example, with coronavirus, we're trying to enhance your immune system in order to make it fight the coronavirus. Where with cancer, we have to actually go outside of your own immune system, supply a different battleground against the cancer cells. And again, these are cells that are dividing too quickly and burrowing in or causing damage to the good tissue that surrounds them. This surgery up here, I always like to point this one out. This surgery up here is for lung cancer. So many people, I mean, I don't like to show gross surgeries or big scars that maybe would bother people for non-preventable surgeries like breast cancer, but lung cancer is a pretty preventable disease. Don't smoke, guys. Don't do it. This is the surgery. Now, you might wonder why they went from the back instead of from the front. Well, if you go through the front, you know you have a heart in the way. And so the surgery is much more dangerous. So when they do a surgery to remove your lung, they have to actually cut in along the ribs. And so that's why they have this crooked scar. And then they have to use a rib spreader, breaking your ribs in order to get in and remove the lung tissue. Another thing to think about is that while you're healing from lung surgery, there is no rest for the lung. The lung has to keep breathing, even though you just had surgery on it. It's a pretty rough surgery. Definitely not something that I would want any of you guys to go through. Please don't smoke. Prevent the cancers that are preventable. Okay.